Hey there folks, and welcome to the second video for Chapter 27, Reproduction and Embryonic Development. In this video we're going to get into the first actual stages of fertilization and post-fertilization embryonic development. So what are those first stages and what do they look like? Well, first let's start with fertilization. When a sperm successfully makes contact with an ovum, with an egg, it has to be the right sperm. There's research now that suggests that there are certain changes in the proteins on the surface of a sperm that have to take place while it is traveling to the egg for the egg to be receptive to the sperm. If that egg accepts the sperm, however, this is what happens. Inside the sperm, there is a vesicle, a small membrane-bound little packet called the acrosome, which contains enzymes that help the sperm digest its way through the outer layer of the egg so that the nucleus, here in green, can eventually be injected into the egg cytoplasm, at which point the rest of the sperm can more or less just fall away, or rather, usually it fuses to the cell surface, to the egg surface, and the DNA inside the sperm nucleus is combined with the DNA inside the egg's nucleus. From this point forward, we are not dealing with a separate egg and a separate sperm. We are dealing with one zygote, a fertilized egg. After fertilization, let's start here. Before fertilization, this is what an egg looks like. You see the egg's cellular membrane, and right in here, you can see its nucleus. The moment an egg becomes fertilized, though, it becomes a zygote, something called a fertilization membrane starts to balloon out from around the egg. This is to protect the egg from any further sperm interfering, and it also helps protect the brand new cells that are going to start forming in the next few hours after fertilization. Notice after the next few hours, once a zygote is formed and that egg is fertilized, the zygote starts to undergo something called cleavage, which is a fairly rapid form of mitosis to make new cells and start development. We go from 2 to 4 to 8 cells to 16 cells. We go to a solid bundle of cells called a morula, and eventually, after a couple of days, we reach a stage called the blastula, which is a hollow ball of cells, and the space inside the blastula is called a blastocele. After we reach the stage of about the late blastula, where it looks like this, kind of like a beach ball made out of cells, hollow on the inside, eventually there's going to be a region, usually around the bottom, it depends on how the ball bounces, where a layer of cells is going to dive inward and form a pocket. At this point, we have gone from blastula to gastrula, and this is a very important stage of development. Gastrulation, as this is called, is the stage where in embryonic development we first start to see specialization of cells and the formation of specific tissues. At this point, especially by late gastrula, we start to see the formation of tissues that will determine which cells eventually become specific parts of the adult body later on in life. Specifically, in the gastrula, there are 3 different layers. On the outside, there is the ectoderm. Derm means skin. Ecto means outermost, so it is the outermost layer. In the middle, there is the mesoderm, meso meaning middle, and on the inside, there is the endoderm. The cells that end up in the ectoderm will eventually become things like the brain, the nervous system, and the skin. From the mesoderm, we get our muscles and our skeleton, as well as our heart and our kidneys. And from the endoderm, we get the rest of our vital organs, our lungs, our liver, our intestines, our stomach, you name it. All right, except for the reproductive system. The reproductive system does come from the mesoderm. Make sure we get that. While this development is going on, there's a whole new suite of hormones that become involved, goodness yes, and also a whole new suite of genes. These genes control things like induction, which is when one group of cells starts influencing the development of another group of cells next to it, and hormone concentrations between the cells is part of how this is communicated. So say it is how specializing cells that are going to become part of the central nervous system can influence the likewise development of cells around them. We also have apoptosis, which is the scheduled death of cells. This is, for instance, in embryonic development, how we go from appendages that look like fins, like you might find on a dolphin, to the separate, delicate fingers you get in human beings. Eventually, well, at one point, we all had that same flipper-like structure. But as humans go through embryonic development, eventually, apoptosis causes furrows to open up between the bones of the flipper and eventually form fingers instead. The hormones that help this communication happen are managed by sets of genes called homeotic genes, and these genes sometimes occur in larger groups called homeoboxes, homeo meaning the same. Homeotic genes and homeoboxes are genes that are fairly consistent even between very different species, in this case between a human and a fruit fly, and they manage the development of fairly similar structures, or at least similar areas of the body in very different types of animals. So in this case, we have a system of homeoboxes, the acronym for which is LAB in the fly, and a whole suite of them, of Hox genes in the human that have to do with the development of things on the head. However, there's also the area around the head and neck that is determined by DFD in the fly, and across a very similar set of Hox genes in the human, and so on and so forth, for the trunk, right, the torso of the human, and the limbs, versus similar areas on the fly body. It's not that they necessarily are responsible for creating exactly the same appendages, but they are responsible for correct development in similar and related areas of the body. And of course, the more similar and closer related the different species are that you happen to look at these for, the more similar those appendages and those structures become. Alright folks, this is going to wrap it up for the second video for Chapter 27. I'll see you next time. Bye folks.